Very good afternoon everyone. In our previous class, we have discussed why classification is needed, and we have also seen different kinds of classifications which were given by different kinds of scientists. And we have also seen an important classification, 5-kingdom classification, which was given by R.H. Whittaker. The first one is Monera, the second one is Protista, and third is Fungi, fourth is Plantae, and fifth is Animalia. In this today's lecture, we are going to discuss the first kingdom of classification. Monera. Before going into this topic, I want to ask you a few questions. Tell me one thing, in last 24 hours, how many different kinds of organisms did you see? Humans and different kinds of animals like cow, buffalo, and different kinds of plants and different kinds of insects like cockroach, mosquitoes, houseflies, etc. Right? But there are some organisms which cannot be seen with our naked eyes. They are known as microorganisms. Microorganisms. Micro means small. Organisms means below. Living things. So if I take a spoon of sand from a garden, it has some thousands and thousands of microorganisms in it. If I take a drop of water from a pond, just a drop, it has some hundreds and hundreds of microorganisms in it. Right? Just now you are sitting in this classroom, and on your hands and on your face, there are hundreds and hundreds of microorganisms on it. You won't believe me, even inside your body there are hundreds of microorganisms. All these microorganisms cannot be seen with our naked eyes. One such microorganism is bacteria. So all bacteria are placed under this category, this kingdom, right? And one more question. What do you think? Which organisms appeared first on earth? Humans, plants, or dinosaurs? None of them. The first organisms which appeared on earth are very simple. Like their organization is very simple. So the organisms which appeared first on earth are known as primitive organisms. Which means primitive means oldest. All the oldest organisms means like all the earliest organisms which appeared first on the earth are placed under this category Monera. We have seen two things. Monera are bacteria and Monera are primitive organisms. Remember this term, primitive, which means the oldest. Right? And this, coming to characteristics, first characteristic is unicellular. Let's break this term. Uni means single. Cellular means cell. These are unicellular organisms, means they have only single cell. When we take a human body, for to think, we have brain cells. For locomotion, we have muscle cells. For response to stimuli, we have nerve cells, etc. For every function, we have different kinds of functions. But for them, they have only one cell. With the only one cell, they will do reproduction, they will do digestion, they will do locomotion, everything. All the functions will happen with only single cell. So all monerans are unicellular organisms. The second one is all prokaryotes. Let's break this term down. Pro means simple or primitive. Carry on. Before going into this term, I want to ask you something. Which part of cell controls all the activities? Nucleus. Carry on means nucleus. Primitive nucleus. So let's take a cell, a plant cell or an animal cell. So they have a nucleus which is surrounded by a membrane called nuclear membrane. This is the reason why this nucleus is a true nucleus. And these organisms are known as eukaryotes. U means true, karyon means nucleus, true nucleus. The organisms like plants, animals, like except Monera, the remaining all kingdoms comes under eukaryotes. Coming to prokaryotes. So this is a prokaryotic cell and it's a nucleus. This nucleus is not covered by any of the membrane. So this is a false nucleus. It's not a true nucleus. Not a true nucleus. It's a simple nucleus. That's the basic difference. In upcoming classes, like in this unit of cell, we will learn much deeper differences between prokaryotes and eukaryotes. For now, just remember that prokaryotes doesn't have any nuclear membrane surrounding the nucleus. But for eukaryotes, their nucleus is surrounded by a membrane called nuclear membrane. Right? That's the reason why eukaryotes are known as organisms with true nucleus. So all the prokaryotes are included in this category, Monera. Third, cell wall. Cell wall. For suppose, let's take a balloon which is filled with water. What does this outer part do? It protects. It protects from outer external environment, right? And it gives a shape. It gives a particular shape for this liquid. And also, it keeps all the content intact, it's closed. For suppose, imagine if there is no outer part, what will happen? It will burst. In the same way, the prokaryotic cell is enveloped by a membrane called cell wall. It protects from external environment and it gives a particular shape and it keeps all the cellular content intact. If there is no cell wall, the cell will burst. In prokaryotes, the cell wall is made up of peptidoglycan. Remember this term. In Monera or bacteria, the cell wall is made up of peptidoglycan. And in plants, the cell wall is made up of cellulose. And in fungi, it is made up of chitin. So, three different terms. In Monera, it is made up of peptidoglycan. And in plants, it is made up of cellulose. And in fungi, it is made up of chitin. So, the cell wall of Monera is peptidoglycan. Where do these organisms live? Everywhere. They are present in soil, they are present in air, they are present in water, they are present everywhere. They are even present in extreme conditions like extreme salty conditions, extreme like whatever conditions it is, extreme heat, extreme salt, extreme acidic conditions, they are present each and everywhere. So we can also call it as cosmopolitan. So the next one, we have finished prokaryotes, cell wall, habitat and reproduction. What do you think? How do bacteria reproduce? Do they have any seed, or do they have any egg, or do they have any reproductive gametes or something? No, they don't have anything. They just have only one cell. That cell, they divide, they grow, and they just divide. Binary. By binary fission. The reproduction happens by binary fission. Binary means two. Fission means divide. It divides into two. That's why it is known as binary fission. So let's take, this is a bacterial cell with DNA. This grows and then the DNA duplicates. And this one becomes two. And this two becomes four. And this 4 becomes 8. So 1 becomes 2, 2 becomes 4, 4 becomes 8, and etc. and n number. This is very fast. This system is very fast. This is the reason why they are present in very more number, and also these are present everywhere just because of this. And there are some other reproductive mechanisms for Monera, like asexual reproduction through spores and genetic recombination by conjugation, transformation and transduction. Like we will take a special class for them, and I will explain you in a very detailed way. So in most of the bacteria, the reproduction happens by binary fission. And the sixth one is nutrition. Nutrition. We are living organisms. We need some source of energy to work. In order to breathe, we need something, an energy. We need some energy. In order to digest our food, we need something. In order to walk, we need something. We need something to work. And that something is food. And for them, how do they get food? There are different kinds of nutrition habits in Monera. The first one is autotrophic. And heterotrophs. These autotrophs are the organisms which can prepare their own food, like a chef. They act like a chef. They prepares their own food. Prepares own food. But these heterotrophs, they are like us. They cannot prepare their own food. Cannot prepare own food. Autotrophs are the organisms which can prepare their own food, like plants, etc. And heterotrophs are the organisms which cannot prepare their own food. And again, in autotrophs there are photosynthetic autotrophs and chemoautotrophs. Photosynthetic autotrophs are the organism which uses sunlight. They use sunlight in photosynthesis. Like they use CO2 and they use sunlight, and with the help of chlorophyll they will give the organic material and oxygen. But coming to chemotrophs, they don't use sunlight because at some places the sunlight cannot pass through. If we take the deepest part of ocean, the sunlight cannot go there. So or in shady regions or in dark regions, the sunlight cannot come and they cannot come into that place, right? That's the reason why they developed a different kind of mechanism. Here, instead of sunlight, they use chemicals. Use energy which is released by inorganic or organic chemicals. Photosynthetic autotrophs are the autotrophs which can make their own food using sunlight, but chemotrophs, they don't have sunlight. They don't have source of sunlight. That's the reason why they use chemicals. Like they break the chemicals and they use that energy. And coming to heterotrophs, these are two types: saprophytes and parasitic. Saprophytes, they depend on, they get their food from dead and decay. And parasitic, we know, parasitic organisms, they depend on host and they take the host nutrients. Sometimes, in some conditions, it might kill or it might affect the host also. So, saprophytic animals, so heterotrophs are divided into two types: saprophytes and parasitic. Saprophytes means they depend on dead and decay. They doesn't depend on living organisms. They depend on dead and decay. Because of this, the bacteria are known as decomposers. Just because of this. So coming to nutrition, there are two types: autotrophic and heterotrophic. And autotrophic are the organisms which can prepare their own food. Heterotrophs, they cannot prepare their own food. And again, autotrophs are divided into two types: photosynthetic and chemotrophs. Photosynthetic autotrophs are the autotrophs which can use the sunlight and prepare their own food. But at some places, at the deepest parts of ocean or in dark shade regions, they don't have the ability of sunlight. So what they do? They break the chemical energy like nitrogen or sulfur, iron, methane, etc. They break those chemicals and they get their energy, right? Coming to heterotrophs, they cannot prepare their own food, and it is divided into saprophytes and parasitic. Saprophytes, they depend on dead and decay. They doesn't depend on living organisms. This is the reason why they are known as decomposers. Most of the bacteria of Monera are heterotrophs. And again, in heterotrophs, most of the bacteria are saprophytes. Now let's go to the classification. Bacteria are divided into 4 groups based on shape. Based on shape, they are divided into 4. Coccus, Bacillus, Spiralium, and Vibrio. So coccus means these bacteria are in the shape of the moon, means circle. These are round shape. These are bacillus of the organisms which are rod shaped. How does the rod look? It's like an elongated one, right? Like this. Rod shape. Spiralium, they have the spiral shape like this. Spiral shape. Vibro, these organisms are like comma shape, comma, like half moon. Or comma, like this. So, according to the shape, the bacteria are divided into 4 groups: coccus, bacillus, spiralium, and vibro. The coccus are round shaped or in circular in shape, like a moon, and the bacillus are rod shaped, elongated rod shaped. And most of the Monera or bacteria are bacillus. One good example is lactobacillus. And for coccus, we can take streptococcus. And coming to spiralium, all the spiralium species. Vibro, these are like one good example is cholera. Cholera is in vibro shape. Cholera is because of bacteria. So it is in vibro shape, means like a comma, like or half moon. Spiralium are like spirals. So this is the classification according to the shape: coccus, bacillus, spiralium, and vibro. Coccus are round shape, example streptococcus, pneumonia, etc. And the bacillus, the most number of bacteria are in bacillus shape, and they are rod shape, example lactobacillus. And coming to spiralium, they are spiral in shape, and all the spiralium species belong to this. And vibro, it's like a half moon and or comma shape. Vibro cholera is a good example for vibro species. So this is according to the shape. The real classification starts here. So what do you think? Do all bacteria look alike? No. Just like humans are not alike, or animals are not alike, or plants are not alike, bacteria are also different. There are different kinds of bacteria. Some they will live at extreme salty conditions, some they will live at extreme heat conditions, and some they will live at extreme acidic conditions, and some might live at normal conditions. And classification. Archaebacteria. So I will compare them so that we can easily remember them and it will be useful for us, and also we can easily understand eubacteria. Bacteria are divided into 2 main classes: archaebacteria and eubacteria. So archaebacteria, archae means ancient. These are ancient bacteria. Like these are the first organisms which appeared first on earth. Ancient bacteria, most primitive organisms. What is the meaning of primitive? I have told you before. Primitive means the oldest organisms which appeared first on earth. And these are extremophiles. Extremophiles. Extreme means extreme conditions. Phile means loving. They love extreme conditions. So there are 3 different types of classes in it again. Methanogens, halophiles, and acidophiles. So methanogens are the organisms which convert the carbon dioxide into methane gas. So they convert CO2 into methane. So these are present inside the digestive system of cows and cattle. Example, these are present inside cattle, rumen of cattle. This is the reason why the cow dung has methane gas. So this gas can be converted into gobar gas, which has numerous amount of so much importance for it. In these days we can use it as a manure, we can use it as a fuel, we can use it as a gas. There are many uses about it. We can learn about this thing in the next year. In the last year, applications of microbes. So methanogens are the bacteria which convert the carbon dioxide into methane. These are present mainly inside the rumen of the cattle, inside the cattle digestive system. And halophiles. Halo means salt. Phile means loving. These are salt-loving organisms. These are present in the extreme salty conditions. So if I take you to extreme salty conditions or extreme salty water, can you stay there? No, nobody can stay there. Because the pH is very, very less there. And coming to thermoacidophiles. Thermo means heat. Acidic. Acid means acid. Phile means love. They love extreme heat with acid conditions. Example, they are present in hot springs. So these are present in the extreme heat, extreme heat and acidic conditions. So Archaebacteria are the ancient bacteria, and they are the most primitive organisms, and they are extremophiles. They love extreme conditions. It's divided into methanogens, halophiles, and thermoacidophiles. Methanogens, they convert carbon dioxide into methane gas, and halophiles, they are salt-loving, and thermoacidophiles, they love extreme acidic conditions and heat conditions. So one more thing. The main difference between Archaebacteria and Eubacteria is their cell wall. It has peptidoglycan, we have discussed earlier. It has peptidoglycan, but this doesn't have peptidoglycan. Peptidoglycan is absent here. Instead of that, they have pseudo peptidoglycan. This is a cell wall that is surrounded the outer part of the cell. This is the reason why they survive in the extreme conditions. Because these are most primitive organisms, they are also known as living fossils. And the eubacteria. Coming to our second one, eubacteria. This U means true. True bacteria. These are true bacteria. Here we are discussing about ancient bacteria. Here we are discussing about the true and the actual bacteria. This is mainly divided into. There are different kinds of bacteria. Like for us, two main important things are important here: cyanobacteria and mycoplasma. Cyanobacteria. These bacteria are also known as blue-green algae. Blue-green algae. In the beginning, like few hundreds of years back, in the beginning when the classification was happening, the people thought this cyanobacteria as an algae because of its color and because of its photosynthetic nature. And after some time, they classified and they kept it in this group because they are prokaryotes. And they have, the main thing in them is photosynthesis. They have chlorophyll A, which helps in photosynthesis. Oxygenic. This photosynthesis is oxygenic, which means they release oxygen after their photosynthesis. And they have a specialized organ called heterocyst, which helps in nitrogen fixation. N2 fixation. Example for this cyanobacteria: nostoc and anabina. Coming to mycoplasma, there are three important parts. The first one, these are the smallest living organisms. These are the smallest living organisms. The second one is they don't have cell wall. Cell wall is absent. And they can change their shapes. Because they don't have cell wall, they can change their shapes. Change of shapes. Monera are divided into archaebacteria and eubacteria. Archaebacteria means the oldest bacteria, the ancient bacteria. These are the most primitive ones. That's why they are known as living fossils. Still, they are present in the present living world. That's the reason why they are known as living fossils. And these are extremophiles, which love extreme conditions. Methanogens, halophiles, and thermoacidophiles. Methanogens means they convert the carbon dioxide into methane gas. And halophiles, they are present in extreme salty conditions. And thermoacidophiles, these are present in hot spring conditions. Like they love heat and acidic conditions. And the basic difference between archaebacteria and eubacteria is their cell wall doesn't have peptidoglycan. But coming to eubacteria, they have the peptidoglycan. They are the true bacteria. Eubacteria is of many kinds. In that, two important bacteria are cyanobacteria and mycoplasma. And cyanobacteria, blue-green algae. In the beginning of classification, people thought that because of their photosynthetic nature and green in color, they thought cyanobacteria as an algae. And after some time, they got to know that they are prokaryotes. So after that, they placed all the prokaryotes into this category, Monera. They have chlorophyll pigment A, which helps in photosynthesis, and they release oxygen after their photosynthesis, just like their plants. And they have a specialized organs like heterocyst in the roots, which helps in nitrogen fixation, which are present in nostoc and anabina. And coming to mycoplasma, these are the smallest living organisms, and they don't have any cell wall. That's the reason why they can change into any of the shape. So that's it for today's class. In the next class, we will discuss about the reproduction in bacteria in a much deeper way. Like we have different kinds of genetic recombination like conjugation, transformation and translation. We'll discuss about them. Thank you.